DNA, deoxyribonucleic acid. DNA is a part of us that stores all of our genetic codes and the schematics, i.e. mathematics of our cell activity. DNA is a 6 billion letter coding that expresses the assembling instructions for everything that you are, representing the balancedness of you and life itself. Each cell within you has 46 chromosomes, each containing a single colossal DNA molecule. Nucleic acid represents the fourth major group of biological molecules, as the first three are 1 carbon, 2 lipids, 3 protein, and 4 nucleic acid. And as structures, these are called polymers, which means that each one is made up of many small repeating molecular units. In DNA, these are called nucleotides. Combine them to form a polynucleotide. 3 forces unite to form a nucleotide. 1, a 5-carbon sugar molecule. 2, a phosphate group. And 3, one of 4 nitrogen bases. The first part of the name DNA derives from the deoxyribose, the sugar molecule. But there are other parts of these gene codes that makes you who you are, as these other parts are based around the 4 nitrogenous bases. DNA does not exist as a single polynucleotide molecule. DNA exists as a pair of molecules, similar to a twisting staircase, as of a double helix. And with this divine structure comes a support system. In DNA, the sugars and phosphates combine to form a twin backbone. The sugar phosphate runs down each side of the twin helix, but chemically in opposite direction. Balancedness. A strand gets to the top of the first phosphate, connecting to the sugar molecule's 5th carbon, and ending where the next phosphate will go, with a free end at the sugar's 3rd carbon. This creates a flow called 5' to 3'. And on the other side of this double helix is what one would call the 3' to 5' flow, beginning up top with a free end at the sugar's 3rd carbon, and the phosphate connects to the sugar's 5th carbon all the way down. These 2 chains are linked via the nitrogenous bases, so one must keep in mind that if a nucleotide is adenine base only, then thymine is its only counterpart. And the same goes for guanine. Cytosine is its only counterpart. These bonded nitrogenous combinations are called base pairs. The GC pair has 3 hydrogen bonds, making it somewhat stronger than the AT pairing, which has 2 hydrogen bonds. DNA is the genetic instruction guide for life and its configurations, i.e. calculations. DNA is the instructions for an organism's development and reproduction, or each cell's development and reproduction. DNA is even the factor when it comes to death. As to say, one of the main roles of DNA in the cell is the long-term storing of information. DNA is vital to nature and all living beings. DNA is life, but it still as a reflection of spiritual DNA. Our spiritual DNA reflects our physical DNA, and as a being, many are using their physical material selves as one of the tools in order to get better acquainted and more in touch with the spiritual DNA. Our physical selves is the result of the activity of material DNA, which is the result of our spiritual DNA. And when you look at the activity of physical DNA with the mind's eye, the activity, one should be able to see some of the rhythms and dance moves of the universe, similar to the rhythms and dance moves of man. Man, you are the you in universe. The you in I is the I in you in universe. So when you look at everyday people dying as they live life, you will see them connecting with different types of people and elements. You will see them with jobs and occupations, particular functions, calculations, and configurations. People coming together for a common cause or mission, similar to producing an offspring or twins. We create, we duplicate, we reflect, we replicate, we reproduce, even our own cell tissue. For example, many of the repair mechanisms within us utilize the undamaged complementary DNA tape as a template to properly correct the lesion tape, even if both gags of the double-strand DNA are broken. Our inner mechanics will solve this problem by removing the information from the DNA sequence present on the other strand of DNA that replicates in a fork table or is present on the homologous chromosome. We reproduce, we repair things, we duplicate and replicate We are DNA. AND RNA. RNA, like DNA, has a sugar phosphate backbone connected to a nucleotide base, but they are different. Unlike DNA, which is a double-stranded molecule, RNA is a single-stranded molecule, and the sugar in RNA is called ribose, which has one more oxygen atom than deoxyribose. And RNA contains uracil instead of thymine. In RNA, the fourth base nucleotide is uracil in place of thymine, for which thymine is the fourth base nucleotide of DNA, meaning adenine connects with uracil instead of thymine. There are some RNA molecules that play an active role in cells by breeding biological reactions, controlling gene expression, or sensing and communicating responses to cellular signals. One of these active processes is protein synthesis, a universal function in which RNA molecules direct the assembly of proteins to its destination. This is an organization because without DNA, RNA cannot do its job properly, which means the ribosome zone cannot properly do its job. And without this, we cannot properly produce proteins. Replication. Every cell in the body holds the ingredients to the same DNA codes that derive from the source, i.e., the parental DNA codes. And these cells will copy itself over and over again throughout a lifespan, but only using half of the original DNA strand template each time to breed anew. And in order for this freaky process to take place, we must first unzip our genes. some may call the helicase. The enzyme helicase breaks down the hydrogen bonds between the nitrogen bases that holds the two strands together, unzipping the DNA molecule. And where this protein begins is the location of the replication fork. The above strand is the leading strand, and the below strand is the lagging strand. These two strands are now templates that go in opposite directions. Due to their chemical fixture. This is in order to balance out two complementary DNA strands. And since the lagging strand flows in the opposite direction as the leading strand, it has to be duplicated in a series of segments. The RNA primase lays down an occasional RNA primer to first give the DNA polymerase a place to begin, then works backwards along the strand. This is done in a bunch of individual segments, each starting with an RNA primer called the OKAZAKI fragment. OKAZAKI fragments form during DNA replication, functioning as the building blocks for concise DNA synthesizing of the lagging strand. This allows a new type of DNA polymerase to go back through and replace all the RNA primers. And then at last we have the enzyme called the DNA ligase to bring all the fragments together.
The DNA ligase.
But of course, DNA has a double-stranded backbone. And during the late 1900s, researchers believed that the replication of both strands occurred in continuous form. Then they came across what is called the DNA polymerase, which occurs in 5' to 3' activity, as they had yet to be exposed to a 3' to 5' activity. Over time, a hypothesis was birthed. This hypothesis was that one of these strands has to function in a discontinuous pursuit, and this strand was labeled the lagging strand, as opposed to the leading strand that functions continuously. And after putting this hypothesis to the test, the presence of small DNA fragments gave notion that this hypothesis was accurate. Synthesis of one of these strands occurs in a 3' to 5' motion, which expresses this continuity. And the longer the continuous strand, which is the leading strand, increases. This leads to each end of the lagging strand, which is the discontinuing strand, joining to form a continuous strand. Reiji OKAZAKI was from Japan, the relative of China. These two are fond of the yin and yang, balance, similar to the number 8, which is a lucky number embraced by both Japan and China. In the eyes of many, Okazaki was someone who had an important purpose to fulfill, stemming from birth, as birth is brought through DNA. That which is a double helix, having a twin backbone, working in a cycle called 5 to 3 prime and 3 to 5 prime. 3 plus 5 equals 8, and 5 plus 3 equals 8. The infinite whole of spiritual and soul DNA exposes itself in the image and activity of physical DNA, which is the codes of human genetics, has to say. Molecular mechanics and DNA was the work of Reiji Okazaki, as well as Reiji Okazaki was the work of molecular mechanics and DNA.
We are DNA, AND, RNA.